pretty good stab at these first two learning objectives that we wanted, or these first two parts of the, but now I want to separate. So what I've done is internal cycling up to now. All of these are things that internally cycle within the ecosystem. So this isn't new nitrogen. There's no nitrogen. It's all the same, and it's just being processed every year continuously through this system. But there are some additions, sources and losses, and those turn out to be really, really important in environmental biology. So for time immemorial, there's been a couple of primary sources of novel nitrogen to ecosystem. The first is biological fixation. So basically, one rule you have is vertebrates suck at biochemistry, plants get a lot better at it, and then the microbial and fungal community are the experts. And so there's lots of reactions that we can't do, but the microbial community can do. And one of the big ones is nitrogen fixation. So nitrogenase is one of the most amazing enzymes. Don't expect you to know this, but these numbers is how you can go through all of these different subunits of the enzyme and do this reaction that's incredibly difficult because you're taking this majorly bonded N2 molecule and eventually coming up with ammonia right there, which will then be in equilibrium with ammonium in the soil. There's two different categories of this kind of fixation in terrestrial systems. The first are symbioses between plants and bacteria. And that's what you see here, these little nodules right here. That's plant tissue that has enclosed nostoc, in this case, a bacterium, in a mutualism. And the benefit to the plant is these bacteria in there will do this reaction and provide a nitrogen source. From the bacteria's point of view, this is a really nice place. It's temperature controlled. It's like an air-conditioned sort of pad and stuff, and it's getting a constant carbon source. So the plant's providing photosynthate to it. There are free-living versions of these bacteria that just live in soil and do nitrogen fixation without a symbiosis. There's nitrogen-fixing cyanobacteria and bacteria in ocean systems, in freshwater systems, in lakes, sediments, etc. So it's been a major source of N to the nitrogen cycle. So we can add this to our thinking. And that added part is, we're going to call it approximately 0 per mil. And the reason I put the approximate there is there have been lots of experiments where you look at nitrogen-fixing plants, and it doesn't come out as 0. It comes out further away from 0. Just as a brain exercise, can you think of any reasons why a nitrogen-fixing plant wouldn't be 0 if all that nitrogen that's coming from the bacteria is 0? So I just told you that all this nitrogen that's fixed this way, it's 0 per mil. There's no fractionation, so it looks just like our standard. Atmospheric N2 is our standard. The bacteria doesn't fractionate it at all. So all the nitrogen that this plant in this symbiosis is getting should be 0. But when we look at those nitrogen-fixing plants, they can vary between 3, 6, up to 6 or 7 per mil. Any ideas why? John?
16 ATP.
Oh, it's wrong? It's not balanced?
No, that's correct. It's expensive.
Yeah. So it's the plant's not getting all of its nitrogen from the mutualism. It can pick it up itself. And John's exactly right. Not in all plant species, but often this is facultative, and it's totally depending upon that energy cost, that 16 ATP that it costs. So if you fertilize the nitrogen, if you have like a black locust growing in your yard, and you fertilize your yard every year, it's probably not doing any nitrogen fixation because it's saying, thank you, I have all the nitrogen I need. I don't need to pay these little vagabonds to make my nitrogen. The other, which I'll get into later, is we don't like to admit this, but there's within plant fractionations that have been documented. And I'll talk to you about them, and then I'll tell you how to cautiously minimize those if you're going to work with N in plants. Okay? The other place that a lot of nitrogen came to ecosystems naturally is from deposition. And we'll get into it, but pre-us mangling the processes of Earth, there were a couple of primary sources for this. The first was lightning strikes. Lightning strikes are one of the few things that have enough energy to break those strong bonds in N2 and make oxides of nitrogen. And then that would be in the atmosphere, and eventually it would acidify and would come back down and would add nitrogen to ecosystems. In addition to that, we already know from our diagram over here that at least in these two places, we have very reactive forms of nitrogen coming out of the soil. Okay? The N2O and N2 are so stable, they have very long atmospheric lifetimes, and they don't really participate in this. I mean, once it's an N2, its turnover time is in the gazillions of years, and you never see it again. But NO and ammonia don't. They can come back down. So they're going up into the atmosphere and then coming back down in precipitation or in dry deposition. So these were the big sources of nitrogen pre sort of us. So the other one is deposition. And this is one place I'm going to have to give you just a variable, because depending upon what is driving that deposition, the isotopic composition of that deposition varies all over the place, depending upon where it's coming from. Okay? Now, since humans came along, we started to do stuff. First thing we did is we greatly augmented the degree of this biological fixation. So in agriculture, we said, you know what a cool thing would be is use agricultural plants that are doing the symbiosis so we can capture a bunch of nitrogen into our system. So this is the common one in the US. Anybody know what that is? That's soybean. So this is the nitrogen fixing crop. They alternate with corn all through the center part of the country. This has been a huge amount of nitrogen addition to the system. But compared to this, it's tiny. So if you don't know Fritz Haber and Bosch, you should, because it's probably one of the major things that humans have done in their history that have changed all of our lives. So Fritz here, Fritz was German. He was a rah-rah guy in World War I. He headed up the chemical weapons program for the Kaiser in World War I, generated mustard gas and every other nasty thing that they were putting into warfare at that time. Then between the wars, there was a big drive worldwide that the limitation to killing each other was there wasn't enough substrate to make explosives, because nitrate was very rare. You had to get it. There's only a few natural occurring sources of nitrate. And so that's what he was at the Kaiser Wilhelm Chemical Institute in Germany. And they figured it out, how to do this industrially. So doing the same reaction that nitrogenase is doing, they could make fertilizer, basically, out of N2 and hydrogen. Okay? This process fractionates pretty close to zero. Interestingly, Fritz was kind of a guy. He goes from rah-rah chemical weapons guy to between the wars when he's leading up. That's the time when the Nazi party comes to power in Germany. And he's ordered by Adolf Hitler to fire all his Jewish scientists. And he refuses. And he gets fired. And over time, going to meetings, I guess this is why you go to international meetings, he had made a bunch of friends in England at these chemistry meetings, and they smuggled him out to England before World War II started. And then he sort of spoke out against Hitler, all these sorts of things, and then he was offered the first directorship of the Chemical Institute in Israel, and he accepted, but he died on board the ship going there, and he never actually took the position. So really interesting sort of history. But this allowed us to have as much fertilizer as we want to grow as much food as we want. And this was the Green Revolution. So this is the primary driver of the explosion and exponential growth rate of humanity on Earth since World War II. Okay? We can finally feed everybody, that we're going to drive everything to the carrying capacity as soon as we can and then see what happens. And then they'll start funding ecologists more, because we're the only ones who thought about it for long periods of time. So you have, in addition now, this industrial and fixation. And it's about 0 per mil, but this is a number you shouldn't believe, because the fertilizer substrates rapidly change their isotopic composition once they're processed and in the field, because they're being used. They're going through all of that process. Okay? Yeah?
So who was Bosch?
He was another guy at the Institute.
And he just helped out?
I don't know. I don't know how these things work. You know, Watson and Crick.
I thought maybe he had an interesting story too.
You know,
Related to the Bosch.
I think so.
Bosch.
I don't know. No, it's BOSCH, I think. Is that the same?
Yeah, I think he just helped speed up the process. Like he helped commercialize.
Yeah, it's very much attributed to Haber,
Yeah,
but they call it.
came up with the reaction and doing it like commercial.
Yeah. I mean, he won a Nobel Prize, so it must have been pretty good. On the deposition side, that deposition number, we've really messed this up. So we went from these tiny, tiny little inputs from soil emissions, biogenics, lightning strikes, and all this to that, okay? And this has been basically one of the correlates with the Industrial Revolution, and its big problem is it's not a contaminant. So if we want to remove mercury or we want to remove sulfur, it's usually a contaminant in the industrial process. Reactive forms of nitrogen are a byproduct of combustion. So it's making reactive stuff out of N2. When you push the accelerator in your car, there's enough energy in that combustion reaction to take some of the atmospheric N2 and make it into a reactive form. So trying to regulate that is enormously difficult. So things like the Clean Air Act have done amazing things with certain pollution compounds in the atmosphere and have had zero effect on NOx production because it's difficult. It's really hard to sort of control, and then it underpins most economies of most countries. So I was awarded a Presidential Young Scientist Award during Bush's administration, and they invite 80% of the people, and I didn't get invited. And I'm pretty sure it's because I was working on NOx at the time, and George wasn't a big fan of regulations of NOx. All right? So we add to this deposition, and it's now huge. And the losses, we have two. The first loss from an ecosystem, those are our additions, are these gases. So we're losing those gases. And then in the liquid form, we're losing ammonium and nitrate through leaching. And leaching is that ammonium and nitrate is moving down through the soil profile. And if it doesn't get immobilized or taken up by a plant and put back into the system, it's eventually lost into groundwater. Okay? Strictly speaking, leaching as a process does not fractionate, but leaching rate can have a big impact on isotope ratios in an ecosystem. And the reason for that is if you have a system that tons and tons of nitrogen is being leached, it's only going through this process one time, say. If it's a tightly coupling system where each time nitrogen is picked up and recycled again, you're seeing enrichment each time. So you often see relationships between rates of leaching and the average isotope ratio of soils and plants in a system because of that. You're basically having it go through the cycle multiple times. And each time it goes through, it can get more and more enriched. So that is that. So isotopology 2, something entirely different. This will prove to you that Jim stole from me. But now that we know all of this, and we know these rules, we know a couple things. One is that almost all of these processes enrich the remaining substrate. Okay? We know that everything's produced tends to be lighter. So we should have predictions about what should happen. Oops, look at that, I gave the answer. What happens through this profile? So if we have nitrogen here and it's slowly moving down through the profile, we should have a predictable change in isotopic composition in that. And we're going to do that on the board, and you're going to do it just like you did with Jim. All right? So we're going to replicate that on the board, or maybe we'll just talk about it. All right. We'll talk about it instead. So tell me which one you want to do first, isotope ratio or %N? %N. Okay, so if this is the %N of the soil, this is getting deeper into the soil profile, this is more nitrogen, how should that line go? Should I start on my right, my left? Okay, start on my right, and then what does it do? Goes down and does what? Left. So it's going to do something like that. Okay, what about the 15N? basically the reverse image, right? So we're going to have some input. We don't know what it is. It's going to be that deposition or wherever it enters into the ecosystem. And then it's going to go like this down through the soil profile because it's being processed, going through these reactions and becoming heavier and heavier and heavier. And you see that in the field. This is Newton Nadelhofer and Brian Fry. Brian Fry is probably the most famous isotope person who hasn't come here since the very first class, right? Might be. But they basically suggested this back in 1994 for forested ecosystems, that you have a gradual enrichment through the soil profile and a decrease in the amount of soil N. So you'll see this over and over and over in soil systems all over the place. So what we'd like to do is figure out interesting and cool ways that we can sample this system with plants. Did you have a question?
You never get a system where you were talking about a lot of region. You never really get that where it's going to really deeply push down the percent in soil.
We deal with systems like that in Hawaii sometimes. So we're at, we have sites that are 4.5 meters annual precipitation, which gets pretty crazy. It also gets crazy in assessing leaching at all, because how do you sort of do that? But you do see much less processing of N and kind of single trips of nitrogen through the system. So, good question.